As it's already a long day, I'm going to speed up a bit because we want to go to the apéro. I'm happy to discuss all your questions at the apéro. So what are we doing? We are creating new models, the next generation models for next generation drugs, therapies, to better predict how a drug is working in the end in the human during the clinical trials and at the end. So we heard our lifespan, long and healthy life. We want to become old, but we have the fact that there are diseases around that we need treatment for. And the challenge for the treatment to find, we need to have the treatment, and currently there is a big challenge in the drug development. What is shown beautifully in this picture. In this graph, you see the number of drugs that could be brought to the market by spending $1 billion in research. And you see there's a steady decline. Another thing is that it takes about 10 to 15 years from the first screening to have the approved drug. It costs around $2 billion in average, depending on the drug, what you do. And there's a very low success rate. Again, depending on the drug, it's between 3 to 10%. So you see there's a way and a big room for improvement, increasing efficiency, decreasing time in development, and also decreasing costs. To find out where our technology can kick in, we need to understand just shortly how the drug development process is going. So we have initially a lot of compounds, which are just chemicals, which are screened in the preclinical phases, and then narrowed down until they get to the clinical phase, where they are tested inside patients. First in healthy, then in other patient groups, and in the end, we have the post-market approval. So our technology can kick in where we have models available to try to predict if a drug is working or not, and this is in the preclinical phase. Now to understand what is the current status, the current status in the preclinical phase are very basic models. They are so-called Petri dishes, and these Petri dishes, if you imagine, they don't really reassemble the human physiology. The body, the tissue, the composition, it's very basic. But still, they're very helpful to screen out trash molecules. Once they are trashed, we come to animal experiments. And we all know the voting this weekend. And it's actually good that we voted against it, because right now the technology is not yet there. But we are working on it to get there, to remove animal experiments needed in the drug development. Because most of the people think about animal experiments just about the rats, but there are multiple species tested throughout. And the problem is, in the end, still, you can humanize some animals, but still, human pathology, diseases, cannot be modeled in these kind of models. So it's clear, we need a new technology, and the new technology, what we are working on, is called organs on chip. So what is it? So organs on chip is actually the fusion of two disciplines. It's engineering and it's biology. We are using engineering approaches which are used in the microtechnology fabrication processes. This is why it's called Onchip. It has nothing to do with computer chip or something. It's just about this design here has microfluidic channels in, and I have a chip right here. So this is the engineering part, but we're also fusing it with the human biology. So it's important to take the right cells to build up the model. And in the end, we want to create the body-like environment. So we developed at Alveolix the first breathing human lung on chip that can stimulate cells in 3 dimensions. So what do we simulate? We simulate the air-blood barrier inside this chip, and we can also mechanically stimulate them. So here you see actually human cells breathing inside our chip. So what does it allow then? It allows to create disease models. So we are working on diseases like fibrosis, COVID-19, to find models that we can then test the molecules. But what we also do is we are creating healthy models to check side effects. For example, if there's a question about the cancer drug, does it have a side effect on the lung we can test it. And this is what we do already now. So here, you see the classical preclinical testing of a cancer drug with a success rate of 3%. Annually, it's 160 billion spent around the world in the development of cancer drugs, and it takes 15 years. Recent studies show that using organs on chip in the pipeline, in the development, is increasing the productivity about 20 to 40%, leading to an increased success rate of 4.2%. That's not a lot. But this study was only taking into account to use the technology for side effects. But we can also use the technology for finding out new drugs. And if we are combining it, the success rate will go up. Due to this increase in R&D productivity, there is the chance for the industry to make about 64 billion a year and to reduce the development time to 10%. This needs to be proven, but we are on the right track and we are already doing stuff now. So what do? We are already working with pharmaceutical companies to find out if a drug is safe, to help them decide whether they want to move forward with the drug into the animal experiments, or also creating new models to find out if a drug is even working or not. Then we are creating new organs because we have the lung, we want to go to the gut, we want to go to the skin, because all these things together will help us in the future to replace and reduce animal experiments. And another thing what will enable us, this technology, is to create personalized medicine approaches, because we can simulate humans on a chip. Thank you very much.